Welcome to the Chemistry, Biology and Math Revision Hub. Today we're doing the Pearson Edexcel International A-Level Biology Unit 2 for January 2024. Let's begin with the first question. Question 1. Plants contain tissues involved in the transport of substances. The diagram shows a sunflower plant. So this is the sunflower plant. And they say estimate the height of the sunflower using the information in the diagram. Give your answer with appropriate units. So here I used a ruler. Since this is given to us as 1.6, I measure this distance here and then measure that distance there, and I get the ratio between the two. Now my approximations here, I used an online ruler, but the ratio is going to remain the same, so I got 34, and here we got 55. So the ratio is 1 to 1.6176. So the sunflower is going to be that times 1.6, which is 1.6 times 1.6176, giving us 2.59 meters, and that is my answer here. The key thing is to get a ratio of this length to that length, and then you can be able to multiply this with a value you got from the other part, so you get the appropriate answer, and this is rounded off to 3 significant figures. In part B, the photograph shows a structure that would be found in a sunflower plant. So we can see structure X here. This looks like a sieve plate. So down here they say name the tissue that would contain structure X. Since it's a sieve plate, it's going to be contained in the phloem. And next they say describe how two structures in this tissue enable it to carry out its function. Since this is a phloem tissue, it has a hollow tube that is going to act as a channel for the transportation of substances. And then, of course, it has companion cells that can provide ATP for the active transport processes. So I said it contains a sieve plate and a hollow continuous tube. This allows transport of sucrose, which is translocation, to occur. And then it contains companion cells that allow ATP to be provided to the sieve tube for active transport. In part C, the photograph shows some rings of lignin as seen using a microscope. We can see the rings here. They say which of the following tissue contain lignin. Lignin is contained in sclerenchyma as well as xylem, so there is no lignification in phloem tissues. So the answer should be C, 2 and 3 only. The diagram shows a climbing plant that is growing up a bamboo pole. This is the bamboo pole and that is the climbing plant. So just 2 reasons why a climbing plant may have a lower lignin content than a sunflower plant of the same height and diameter. Because this is a climbing plant, it should have some flexibility, and that is why there is low lignin. So I say to be flexible so that it can bend as it wraps itself around the bamboo plant. It also requires less support to be upright since it's already received support from the bamboo plant. So this brings us to the end of question 1. Let's continue to question 2. In question 2, fish are often packaged for transport to markets. The bacteria E. coli and S. aureus are found on the surface of the fish. These bacteria can cause disease in humans. Which structures enable E. coli to stick to the surface of fish? E. coli contains the capsule as well as the pili. These enable it to stick. The flagellum is not for adherence on the surface, so that is out. Wherever we have the flagellum, that answer is going to be wrong, so the only correct answer is going to be a B. In part B, high-density polyethylene, which is HDPE, and polystyrene are 2 examples of oil-based plastics. The photograph shows polystyrene boxes that have been used to transport fish to a fish market. So they say plant-based packaging is more sustainable than oil-based packaging. Explain what is meant by sustainable. Sustainable means something can be maintained for longer periods of time, meaning we can get the raw materials and we can be able to deal with the waste. So I said plant-based materials can be continuously obtained by growing more plants. They can also be carbon neutral, and they're biodegradable, so the waste can be decomposed by bacteria. Next they say, the effect of packaging material on the growth of viable bacteria on the surface of fish was investigated. The number of S. aureus bacteria on 4 groups of fish was determined before each group of fish was placed in a different type of container. The containers of fish were kept at 1 degree Celsius for 1 week. The table shows the means and standard deviations for the decrease in the number of colonies of S. aureus bacteria. So from the table, we have the different packaging materials, as well as the mean values and their attached standard deviations. So here I got the highest and lowest values and compare them to the mean. So in this case, to get this one, I got that minus that, and to get this one, I got that plus that. I did the same for all the data until the last one for polystyrene. And here they say comment on the results of this investigation. In questions asking you to comment, you have to be able to describe what you see in the results. You have to talk about the methodology, but since here we are commenting on the results, there are no methods that are given to us, even if we do not focus on that. Since they've given us standard deviation, we need to talk about the validity of the results, if there is overlap and so on. Looking at these results, the greatest decrease was here with pine wood, and this one here had the least decrease. So here I said there is decrease in the number of bacteria in pine wood, poplar wood, and polystyrene. However, there is no change in the mean number of bacteria with HDPE packaging. You could say mean or just no change in the number. The decrease in bacterial growth shows presence of antimicrobial properties. Pine wood had the most decrease in growth, so it has the most antimicrobial properties. We saw that from the table. The results included standard deviation and there is no overlap, so the difference in the results is significant. I could go on to say that poplar wood had the greatest standard deviation, so its results have the lowest reliability. And again, to remind you, when they say comment, you have to describe. You could explain the results, talk about validity, talk about the methodology, as well as the sample size. So this brings us to the end of question 2. Let's continue to question 3. In question 3, migration is when many individuals of a species move from one geographical region to another. Monarch butterflies migrate from Canada and North America to Mexico each autumn. The photograph shows a cluster of migrating monarch butterflies on a branch of a tree, which is this one here. They say migration is an example of which type of adaptation? This is going to be behavior adaptation, so the answer should be B. It's not anatomical, not physiological, and of course there is no isolation adaptation. And next they say the heterozygosity index for a cluster of 8,000 monarch butterflies was calculated as 0.166. Calculate the number of heterozygotes in this cluster. The number of heterozygotes should be the heterozygosity index times the total number. So this is going to be that times that, giving us 1,328, which is my answer here. In part B, female monarch butterflies lay fertilized eggs on the leaves of milkweed plants. Herbicides can be used by farmers to kill plants such as milkweed. Farmers in the USA have increased the use of glyphosate herbicide since 1999 to kill plants such as milkweed. The graph shows the percentage of fields in the USA containing milkweed plants from 1999 to 2010. So on the vertical axis, we have the percentage of fields containing the milkweed plants, and on the horizontal axis, we have the time in years. Overall, we see as time increases, there is a decrease in the percentage of field containing the milkweed plants. And since these milkweed plants could be used to lay eggs, it means there are going to be fewer butterflies as time goes on. Then they say explain what effect this human activity would have had on populations of monarch butterflies, including their genetic diversity after 2010. Use the information in the graph to support your answer. If we go back to the graph, this is after 2010, so we'll expect this to decrease more. So I say due to use of herbicides, the percentage of the milkweed plants has decreased. So the monarch butterflies will have fewer sites to lay their eggs. So fewer eggs will hatch and fewer butterflies will be formed. This decreases genetic diversity. In part C, the photograph shows a hen, which is a female chicken that has just laid an egg. We can see this is the hen and that is the egg. They say the rate of egg laying by hens is an example of polygenic inheritance. This phenotype shows continuous variation. State what is meant by polygenic inheritance with reference to this phenotype. Remember, they say the rate of egg laying is an example of polygenic inheritance. So in this case, it means the rate of laying eggs is controlled by multiple genes. So I said multiple genes contribute to a specific phenotype, that is polygenic inheritance. For example, many genes work to cause the different rates of egg laying. Next, they say, sketch a leveled graph to show the expected egg laying rate for a population of hens. So because this shows continuous variation, we have to show a graph like that. But on the vertical axis, we have the number of hens, and then on the horizontal axis, we have the rates. So the rates are going to be different or varying, meaning they're going to be different number of hens hatching at different rates. So this brings us to the end of question 3. Let's continue to question 4. In question 4, the photograph shows some pollen tubes growing from pollen taken from a lily flower, as seen using a light microscope. So we can see the pollen tubes. The pollen tube growth rate is 0.2 micrometer per second. How long will it take the pollen tube to grow? 15 millimeters. 15 millimeters is the same as 15 times 10 power 3 micrometer. So if we're looking for the time taken is going to equal to length divided by the speed, which is this in micrometer divided by the speed. I converted this to micrometer because the speed is in micrometer per second, so the units can cancel out properly. So this gave me 75,000 seconds, but there is no 75,000 seconds here, so I have to try and convert it to minutes. Dividing by 60 to convert to minutes, I got 1 to 50 minutes, so C is my answer. State how to calculate the actual length of one of the pollen tubes in the photograph. We know magnification is equal to image size or image length divided by the actual length. So actual length should equal to image length divided by the magnification. So we will use the microscope to find the image length or image size and then divide by the magnification. So I said measure the image size and divide it by the magnification. Remember, from the photograph we have times 100 as the magnification, so I will divide by 100 to get my accurate answer. In part B, the diagram shows a pollen tube that has grown to reach an ovule. So we can see this is the pollen tube growing in there. It has reached the ovule. Fertilization of the female pollen nuclei results in the production of the endosperm tissue. Fertilization of the female gamete results in the production of an embryo. Explain why an embryo cell and an endosperm cell contain different quantities of DNA. The embryo is going to be diploid because one female nucleus and then one male nucleus do fuse. But the endosperm is going to be triploid because the two pollen nuclei fuse with one male nucleus. So here I said the embryo is diploid while the endosperm is triploid. The embryo forms when a haploid male gamete nucleus fuses with a haploid female gamete nucleus. The endosperm forms when a haploid male gamete nucleus fuses with two polar nuclei. In part C, the photograph shows part of a Madonna lily plant, which is this one here. They say an extract made from L. candidum has been used in traditional medicine as a treatment for human skin burns. Describe a drug trial method that a scientist could use to determine if this extract is, number one, safe to use in humans. So here they could do preclinical trials by testing on human skin cells in the lab before transitioning into humans. And then they say more effective than the current skin burn treatment used in a hospital. So here they have to do a double-blind trial with the medicine as well as the current available skin burn treatment. And again, clinical trials, we have phase 1, phase 2, and phase 3, as I wrote here. So I say test the extract on human skin cells in the lab, and then you carry out phase 1 of human trials. You test the extract on a small number of volunteers without skin burns to observe any side effects or toxicity in the body. In phase 2 of the human trials, you test the extract on about 100 to 500 volunteers with skin burns. These are patients. Here, check for side effects and appropriate dosage, because of course we are testing on people who are sick or who have the condition. In phase 3 of the human trials, test the extract on a large number of people with skin burns. This could be over 1,000. And both phase 2 and phase 3 should be double-blind. one group should receive the extract, while the other group receives the current skin burn treatment used in the hospital. So this brings us to the end of question 4. Let's continue to question 5. In question 5, the photograph shows an organelle found in many plant cells as seen using a microscope. So this is the organelle, and we can see this is a chloroplast. They say give the name and function of this organelle. We could see it's a chloroplast because we can see the lamellae as well as the stacks of thylakoids, which make the grana. So here I said it's a chloroplast, and the function is it's a site for photosynthesis. Name the type of microscope used for this photograph. Give one reason for your answer. If we look at this photograph, we can see everything on the inside. We can see the thylakoids, we see the grana, we see even the lamellae connecting the thylakoids. This means they've used a microscope that has great resolution, and this should be a transmission electron microscope. So I say transmission electron microscope because it has high resolution. Resolution helps us to tell things that are so close to each other apart. In part B, the photograph shows a lake partly covered with New Zealand pygmy-weed plants. They say the effect of New Zealand pygmy-weed on the biodiversity of a lake was investigated. Species richness and an index of diversity were determined. State what is meant by species richness. Species richness is the number of different species that are found in a habitat or in a specific area. The number of individuals of different species in this area of the lake was measured before the appearance of the New Zealand pygmy-weed. The table shows the results. So the results were presented in the table, and down here they said, calculate the index of diversity D for this area of the lake using this formula here. And they say, use the table to help you and give your answer to 2 decimal places. So from the table, there was some information that was not filled out. For example, here we need n-1, which is 12 minus 1, giving us 11. And that times that to give us that. Also here we needed that times that to give us that. In here we needed to find the sum or total n, which is going to be 153 if we add everything up. And here, adding everything up gave me that. So substituting the components into the diversity index formula, I got that times capital N minus 1, which is going to be 152, divided by the sum of small n into small n minus 1. And I got 3.85, which is to 2 decimal places as required. And this was my answer. Then they say a year after the appearance of the New Zealand pygmy-weed in this lake, the index of diversity was found to be 1.8. If we compare the diversity index we got here, it's 3.85. It means there was a decrease. This suggests the effect of the New Zealand pygmy-weed on the diversity of the part of the lake. When they say questions about suggest, it means they want your own opinion. What do you think actually happened? So I said, the pygmy-weeds led to a decrease in diversity index. In my opinion was the weed grows on the surface, blocking the penetration of sunlight. So the water plants are not going to photosynthesize properly. And also there is going to be death and decomposition. So this decomposition could be aerobic, using up the oxygen. So more organisms are going to lack oxygen in the water since there is no photosynthesis occurring to replace that oxygen. So they will die and more decomposition will occur. And this is going to lead to a decrease in biodiversity or a decrease in the diversity index. So this brings us to the end of question 5. Let's continue to question 6. In question 6, all living organisms are made of cells. The table shows some structures found in living cells. They say complete the table to show in which type of cells these structures are present or absent. Amyloplasts are found in organisms that do make starch, so they're going to be absent in animal cells because there is no storage. And then in plant cells, they're going to be present and they'll be absent in prokaryotic cells. Circular DNA is going to be present in all. Mitochondria are going to be present in eukaryotes, so this and that, it's going to be lacking here. And the nucleolus is going to be present in organisms that have a nucleus, which is going to be eukaryotes. Then in part B, they say the smallest living cell has a diameter of 0.2 micrometer. The volume of a sphere is calculated using the formula, which is that. Which of the following shows the volume of this cell in standard form? So since they've given us the diameter and we want to find the volume, I need to convert this to radius. Radius is going to be diameter divided by 2, which is 0.1 micrometer. And volume is 4 over 3 times pi times 0.1 power 3, which is roughly 4.2 times 10 power negative 3 micrometer cubed if you use pi in your calculator, or something close to 3.14. In part C, gamete cells are specialized for their function. The photograph shows some sperm cells as seen using a microscope. So we can see they're here. The magnification is times 4000. They say which of the following statements about human sperm cells are correct? They contain the same quantity of DNA as body cells. Body cells are diploid while sperm cells are haploid, so that is wrong. They contain hydrolytic enzymes. That is true, they have the acrosomes that contain the enzymes. The release of energy from starch enables the rotation of the flagellum. They do not contain starch, so the only correct answer is going to be 2 only. And next they say the photograph shows a sperm cell being injected into a human egg cell during the process of in vitro fertilization. So we can see that is occurring. This is a sperm cell being injected into the female egg. Scientists have suggested that one cause of human infertility is the production of egg cells with a zona pellucida thickness greater than 18 micrometer. The table shows the mean thickness of the zona pellucida of fertilized eggs and unfertilized eggs. So we can see the fertilized eggs, the mean is 16.6, but plus or minus 3.2, which could be 13.4 to 19.8. And the unfertilized is going to be 18.9 plus or minus 4, which is 14.9 to 22.9. We can see there is overlap, so the difference within the results is not going to be that significant. However, I have to first focus on the question. So they say sperm cells are released from the male by ejaculation. Another suggested cause of infertility is a longer time between sperm cell ejaculation and the cells reaching an egg cell in the female. This is true because the enzymes from the acrosomes could be released, and by the time the sperm reaches the zona pellucida, there is not much enzymes for digestion to occur, so there is a possibility of this causing infertility. Scientists wish to increase the number of successfully fertilized egg cells by IVF. They have proposed that the technique of injecting recently obtained sperm cells should be used for egg cells with a zona pellucida thicker than 18 micrometer. When IVF is carried out, the sperm is inserted into the egg cell, which is going to overcome the requirements for the acrosomes or the enzymes that are going to digest the zona pellucida. They say evaluate this proposal. Again, when they say evaluate, make a judgment based on the evidence. So here they've given us evidence from the previous page. Here we need to include the pros and the cons of the methods that are going to be used. They say use the information provided in the question and your own knowledge to support your answer. So I said from the table, fertilized eggs have a thinner zona pellucida than unfertilized cells. Thickness of the zona pellucida seems to lower the chances of fertilization. However, there is overlap between the zona pellucida thickness of fertilized and unfertilized egg cells, so the results are not significantly different. We saw that previously. If sperms take longer, or if sperm takes longer to reach the egg cells, they could have depleted their acrosine, so less acrosome reactions occur and the zona pellucida may not be efficiently broken down or digested for sperm to enter, and the chance of fertilization is going to decrease. Using IVF introduces the sperm nucleus into the egg cell, increasing the chances of successful fertilization. Since there is no need for acrosome reactions, IVF can overcome problems associated with sperm with low motility, or you could say associated with low motile sperms. Since I told you in evaluation questions you have to talk about the pros and the cons. The cons here are women have to take a lot of hormonal supplements to cause multiple eggs to mature at once before harvesting them. You should know that these eggs are going to be harvested by surgery. So basically, the lady is given hormonal supplements so that many eggs mature, maybe one period of a month, so that surgery can be carried out to remove the many matured eggs. So this is going to cause some side effects. I say the side effects from these hormones cause concern among some people. And some people have used the advancement in IVF as a stepping stone for genetic engineering in humans, and this may have many ethical concerns. So this brings us to question 6. Let's continue to question 7. In question 7, a change in the base sequence of a gene can give rise to cancer. The graph shows the number of cases of one type of cancer in a human population. On the vertical axis, we have the number of cases of cancer per 100,000 population. And then on the horizontal axis, we have age. They've given us data for males as well as females. And we can see there are more cases of cancer in males at all ages in comparison to females. However, the cases in females increase until the age range 70 to 79, and then we can see they begin to decrease. Also, for both males and females, we have results from between the age 40 to 49. We do not have results for before that. The cases in males are continuously increasing. So they say deduce the effect of age and sex on the number of cases of this cancer. I say it as age increases, the number of cancer cases per 100,000 population increase. There are more cases in males than females at all ages. And then between 70 to 80 years, the number of cases in females decrease, where the number of cases in males continue to increase. The difference between male and female cases could be due to lifestyle and genetics. And if it's due to genetics, it's going to be sex-linked. The reason for saying sex-linked is we have more cases in males at all ages than in females. And next they say a city had a population of 2 million people. 3% of this population were males over the age of 80. Calculate how many of the males over the age of 80 would have this cancer. So taking you to the graph, we can see over the age of 80, we have about 660 people in 100,000 have the cancer. So the population of males is going to be 2 million. So 3 over 100 times 2 million is going to be 60,000 people. These are males above the age of 80. Then from the graph, in 100,000 population, 660 males had cancer. Those are males above 80. How about these males above 80? It's going to be 660 divided by 100,000 times 60,000, giving us 396, and that is going to be your answer. In part B, a sample of tissue was taken from a patient with a type of cancer in order to calculate the mitotic index. The sample was stained before being viewed using a microscope. State why the cells in the sample were stained before being viewed using a microscope in order to calculate the mitotic index. Staining is to ensure that the chromosomes can be visible. And again, remember, when we're calculating the mitotic index, we observe the chromosomes so that we can see which cells are in mitosis and which cells are not. So the chromosomes should be visible. That is why staining is carried out. And next they say describe how the mitotic index of the tissue sample could be determined. Of course, we're using a microscope. We're going to observe the cells. We'll count the number of cells in mitosis. And mitosis includes prophase, metaphase, anaphase, and telophase. And then we count the total number of cells. These include prophase, metaphase, anaphase, telophase, and interphase. And then we divide the cells in mitosis by the total number of cells. We could multiply by 100. Multiplying by 100 gives us a percentage mitotic index for comparison with other data. One treatment for this type of cancer prevents the condensing of chromosomes of cancer cells. In which stage of mitosis do chromosomes start to condense? This is going to be prophase. Next, they say the graph shows the effect of the mitotic index on the percentage survival for people with a type of cancer. On the vertical axis, we have the percentage survival, and down here we have the time after diagnosis. So those who have low mitotic index and those with high mitotic index both have a decrease in the percentage survival, although those with high mitotic index have greater decrease in comparison to those with low mitotic index. So they say explain how preventing the condensing of chromosomes would affect the survival of a patient with this type of cancer. If the chromosomes cannot condense, it means they will not be able to go through all stages of mitosis. So it means cells going through cell division will be decreased, or the cells going through mitosis will be fewer. If fewer cancer cells are going through mitosis, then fewer tumor cells are going to be made, or fewer cancer cells are going to be made. So I said if the chromosomes are not condensed, mitosis is hindered, metaphase is hindered, and anaphase cannot occur, or it will be occurring at a lower pace. This reduces the number of cells in mitosis and thus the mitotic index. So fewer cancer cells will be formed, and this will stop or slow down tumor growth, increasing the chances of survival among patients. So this brings us to the end of question 7. Let's continue to question 8. In question 8, the map shows 6 islands near the coast of California. These are the islands. They say each of these islands has a population of island foxes descended from the California gray fox. Each island fox population has developed different characteristics from the fox population on the other islands. The photograph shows a fox from the population found only on the island of Santa Cruz. In part A, they say which of the following is a term used to describe the fact that this subspecies of island fox is found only on Santa Cruz. It means it's endemic on Santa Cruz, so the answer should be B. Next, they say, explain how isolation caused the formation of 6 fox populations with different characteristics. Use the information in the question to support your answer. So isolation occurred, and now we have 6 different populations. So it could be geographical isolation, leading to reproductive isolation, and then the organisms that are isolated in the different areas are going to be faced with different selection pressures, and they will adapt to those selection pressures differently, and this will lead to the differences that are going to be observed. So I said, due to geographic isolation, there is no gene flow between the isolated individuals, meaning reproductive isolation has occurred. The organisms are also faced with different selection pressures in their respective habitats, so they are adapted differently, and different alleles have been selected for in the different habitats. So their offsprings have gained different alleles, causing the genetic differences among the populations isolated in the different areas. And next I say, explain how molecular evidence could be used to determine which of the other 5 fox populations is the most closely related to the fox population in Santa Cruz. Since we are using molecular evidence to carry out molecular phylogeny, analyze molecules, these could be proteins, DNA, messenger RNA, and so on, that are produced by the foxes from the 5 populations. Those that have the most similarities are going to be the most or more closely related to those in Santa Cruz. In part B, the graph shows the sizes of 2 fox populations from 1994 to 2012. On the vertical axis, we have the number of foxes. So they've given us one in Santa Cruz and the other Santa Catalina. We can see that in both populations, the number of foxes decrease, and then they increase after some time. Then they say 2 main causes for the decrease in island fox populations from 1994 to 2000 were predation by golden eagles and disease transmission from dogs and raccoons. They say calculate the percentage decrease in Santa Cruz fox population from 1994 to 2002. In 1994, we can see it's 1,500, and in 2002, it's 80. So my decrease was 1,500 minus 80, which is 1,420. So the percentage decrease is 1,420 divided by the original times 100, giving me 94.7%. Explain how the Hardy-Weinberg equation can be used to show the change in the frequency of a recessive allele in Santa Cruz population from 1994 to 2000. So the recessive allele is going to be in the homozygous recessive as well as in the heterozygous. So here we need to calculate the frequency of the homozygous recessive, which is going to be Q. And then we could use P plus Q is equal to 1 to find the P. Then we use p squared plus 2pq plus q squared is equal to 1 to find that for the heterozygous. We'll do calculations in 1994 and in 2000, and then we compare the frequencies in 1994 and in 2000 to see if a change has occurred. This should be C. In 2004, a conservation plan was produced to increase the sizes of the Santa Cruz and Santa Catalina fox populations, and then maintain the genetic diversity of Santa Cruz and Santa Catalina fox populations. Explain how conservation could achieve these goals. They want you to use your information in the question and your own knowledge to support your answer. Here we could do captive breeding. We could put the foxes in gazetted areas that are protected. We could give them medication against disease, could protect them against predators, and then give them enough food. That is for increasing the number. And for maintaining genetic diversity, we ensure that we record the mating pairs. So I say to increase the population, captive breeding programs can be carried out. Also, areas can be selected where foxes are protected, provided with food, medication, and vaccination against infection. We could also reduce the number of predators, which are the raccoons and golden eagles, as well as the dogs. Then to maintain genetic diversity, we could use stud books to ensure that inbreeding does not occur, or to ensure that females mate with different partners. This can also be done under captivity. You could do this in captive breeding as well. So this brings us to the end of question 8, as well as to the end of the whole paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.